It is my pleasure to introduce our speaker for today. And this is Professor Shuji Terai from Niigata University. He's professor currently and chairman of the Division of Gastroenterology and Hepatology at the Graduate School of Medical Sciences at Niigata University. And as we discussed before, he also serves as a vice president of the Japanese Society for Regenerative Medicine, as well as auditor of Japanese Society of Gastroenterology, director of the Japan Society of Hepatology as well. And he's also a member of the Exosome Committee and the Gastrointestinal Committee of the International Society for Cell and Gene Therapy, if I'm correct. So his research has advanced significantly the field of liver disease, specifically through all his innovative work on cell therapy, so cell-based therapies and different regenerative approaches to treat specifically liver cirrhosis and to promote liver repair. And today he will present a talk entitled Therapies for Liver Cirrhosis using Allogeneic Mesenchymal Stromal cells and the development of extracellular vesicle-based treatments. So before we begin, just a quick note for the audience. So we will have a Q&A session at the end after the talk, where you can ask your questions. If you prefer, you can also drop your questions in the chat box during the talk, and then we will address them later at the end. And yeah, I think this is it. So Professor Terai, I think that you can start sharing your screen, and feel free to go whenever you're ready.
Okay, and thank you very much to give me a chance to present this nice webinar. My name is Shuji Terai from Niigata University. I would like to share. Okay.
Yes, we can see. We can see now.
Okay. Okay. Hey, today my talk is treatment strategy for liver cirrhosis from mesenchymal stromal cell therapy to realizing the treatment using extracellular vesicle. This is Fegato. For me, is the first time. And this is my COI. And many people knowing the liver have the capacity for the liver regeneration. And this is the partial hepatectomy. A mice model is partial hepatectomy, and 1 day is fat accumulation is occurring 3 days, 7 days. 70% partial hepatectomy, and this is the 7-day past is 70%. And to regeneration, hepatocyte, bile duct, steride cell, and cellular cell regeneration a little bit is a day is a little time lag. And this is checking why fat accumulation is occurring. This is the triglyceride content is increasing, and the DNA synthesis is increasing. And using this system in Japan is living donor liver transplantation was done. And many countries is doing brain death, but is unfortunately in Japan the brain death is around 100 cases. And this is the living donor liver transplantation. Right lobe is to the transplant. And recently, I heard it recently, the MASH is increasing, but it's a normal condition, chronic hepatitis liver cirrhosis condition, hepatitis C virus, hepatitis B virus, and recently metabolic syndrome and MASH is increasing. And this is hepatocellular carcinoma. And in this condition this is the liver cirrhosis condition and decompensated liver cirrhosis condition. Hepatic encephalopathy, esophageal varices, and bleeding and hepatocellular carcinoma is occurring. And this is a life-threatening event. And many people knowing is fibrosis and degeneration interaction. This is a liver cirrhosis condition. And this is like a carrot potato liver, and this condition is liver regeneration is decreasing. And many people have to regulate it. The liver fibrosis is very important. And thinking of the clinically, this is Azan staining. This is hepatitis B virus. This is hepatitis C virus. This is alcohol and meds and MASH. And please comparing, many people knowing is pseudolobular size is much smaller, alcohol and med. And recently, and alcohol, and MASH is increasing. And this is firstly, when I spending a postdoc in NIH, and this is a very exciting finding is coming from the Professor Sis. And this is a leukemia female patient. And this is bone marrow transplantation. And this is XX, and bone marrow transplantation from male, and XY. And this patient is dying and doing autopsy and finding Y chromosome positive hepatocyte is finding. And as that situation, this is a 2000 paper, and no iPSL is not there at the timing. And I myself is thinking it may be possible bone marrow cell may be using it as cell therapy. I come back to Japan and making this model and GFP transgenic mice model and bone marrow cell, and I checking injection from tail vein, and I checking carbon tetrachloride inducted the reverse cirrhosis model, and control is the normal group, and I checking. And this is a GFP positive bone marrow cell, the green cell, and we checking 1 week, 2 weeks, 3 weeks, 4 weeks past. And this is the first. This is just is a very preliminary data. 2001 is we can finding that green cell is depopulated in liver. This is a depopulated GFP positive bone marrow cell depopulated. And this is a really GFP positive cell is a bone marrow cell derived cell or not? That is the question. And anyway, I doing the perfusion and immunohistochemistry and GFP positive cell. And 1 day, 1 week, 4 weeks, and we can finding a GFP positive cell is increasing this model. And I'm sorry. This is I'm sorry. This is just a Japanese story, and we can finding is that this is GFP positive albumin producing cell is increasing. But important point is the other system, and at that time is many people is not acceptable, and because at that time is one part is trans differentiation, other part is cell fusion. And but this is very important model to proceed with the cell therapy. And we also checking that this is the series rate staining is a bone marrow cell infusion. Please compare 1 week. This is 4 weeks after bone marrow cell infusion, and series rate staining showing on 4 weeks past bone marrow cell infusion and liver fibrosis is improving this model. And we checking more. A GFP positive cell is a black color, and the series red color is a red color. And we can finding it. This is please compare. This is 2 weeks. This is 4 weeks. GFP positive cell increasing and red color and liver fibrosis cell is decreasing. And this is very important finding. And I checking the mechanism. And at that time we can finding is MMP9. MMP9 is matrix metalprotease 9 is increasing GFP positive cell is secreted with MMP9. And it may be related to the improving liver fibrosis. And I also doing inside thermography. And this is the bone marrow cell infusion 1 week, and this is more magnified and pre-checking. And this is thermography activity is increasing this model, and this is control. This is bone marrow cell infusion group, and we can finding at the time, and this is a bone marrow cell, and that is all bone marrow cell infusion, and at that time is the GFP positive cell increasing and the liver fibrosis is improving. And the mechanism is somehow related to MMP9. And this is that is our paper is published and to the editorial board is I don't look showing this is the first time is part of the holy grail. Stem cell, hepatic injury, fibrogenetic and repair is very important. We published the transplantation of the bone marrow cell, reducing a carbon tetrachloride inducing liver fibrosis in mice. This is 2004. And we doing is many experiments. Anyway, is we using mesenchymal stromal cell macrophage infusion, and at that time, MMP9 secretion, and sometimes it's protective cell. But it's important find is we can find improving of the liver fibrosis. And I'm a clinician and proceeded the clinical study. And this is the first clinical study, development of the autologous bone marrow cell infusion therapy. And this is the firstly. This is I just started 2003. This is a decompensated liver cirrhosis model. And we getting is a bone marrow cell and the general anesthesis. We getting 400 milliliter bone marrow cell from ileum and to getting mononuclear cell injection. And in this condition, total bilirubin level is under 3.0 and platelet is over 50,000 and cardio and respiratory function is fine. And this is infusion, mononuclear cell infusion. And we doing it. This is my first paper. And after doing this is pre-ABMS therapy, 4 weeks, 24 weeks. Albumin level is improving, child score is improving. And this is please focus is the liver showing irregularity, but is the liver is irregularity is improving. And this patient at that time is albumin level 2.7, but is now this patient still survived, and 19 years passed. And now this patient condition is albumin 3.8. And I doing is a 62 patient in Japan, is a 42 case, and a 20 case in Korea. And this is the child score is after ABA1 therapy is improving. This is control group. And anyway, many people knowing HPV related and alcohol and HIV and HCV and MASH related liver cirrhosis is the lobular size is a little bit different. So is I go into the Korea Yonsei group is doing is ABM therapy and the Yamagata alcohol related and HIV and HCV and MASH related to Okinawa. And this is the Yamagata group data. The patient condition is improving this patient. And this is the Korean group data. And please focus, child pool score after ABM therapy is improving. And portal collagen area is decreasing after 6 months past. And this is a paper from a Korean Yonsei group. And important finding is we also doing liver biopsy. And we doing autologous bone marrow cell infusion. We can finding this. This is our group data, Yamaguchi group data. We can finding a PCNA positive after ABMI therapy is increasing. And this is the Korean group. CK7 positive hepatic progenitor cell is increasing this model. And this is a hepatic progenitor cell, and hepatocyte is a proliferation and improving liver function. And from this clinical study, I can find one proof. And liver cirrhosis patient is tough fiber is a problem. And so in this condition, it is very difficult to liver regeneration. So in this model is improving liver fibrosis and liver regeneration. Endogenous liver regeneration is occurring in human case. And this is a very important clinical finding from our clinical study. And I'm moving to this is the Yamaguchi. This is Japan. This is Yamaguchi. I was born in Yamaguchi, and I moved to the Niigata. And I starting with why ABMS therapy is effective. And I also to develop to the previously autologous, but every time is I doing autologous bone marrow cell infusion. Important point is allogenic mesenchymal stromal cell therapy. More people we can cure and do a therapy. And so I started in the next study. And this is a Niigata castle. And yeah, many people knowing mesenchymal stromal cell. We can getting a mesenchymal stem cell from bone marrow, but also we can getting is medical waste such as adipose tissue, umbilical cord, and mesenchymal stromal cell low antigenicity. And so I think allogeneic mesenchymal stromal cell can be used. And mesenchymal stromal cell, cytokine, chemokine growth factor, exome, and anti-inflammation, angiogenesis, antioxidant, antifibrosis. And this is I doing is the autologous bone marrow cell infusion therapy started at 2003. Starting time is the basic study 2000. I moved to the 2015 to the Niigata University, and I doing is a clinical trial at 2017. And the important thing is what was occurring, and so I doing a basic study. And thinking again, in bone marrow cell, this is bone marrow. This is mesenchymal stromal cell is exists, but it's a hematopoietic stem cell. Macrophage is also included. And which cell is the most effective to the therapy? So I doing this experiment. This is a carbon tetrachloride injection, 8 weeks, and infusion and mesenchymal stromal cell and bone marrow cell, derived macrophage, and half and half. 50%, 50% mesenchymal stromal cell macrophage. And we checking is this is the liver serous rate staining and liver fibrosis. And please focus here. This is control. Mesenchymal stromal cell. Okay. Liver fibrosis is improving. And macrophage also improving liver fibrosis. And the most effective is half and half, 50%, 50%. Mesenchymal stromal cell macrophage are most effective to improving liver fibrosis. And we also doing a two-photon microscope. Two-photon microscopy is a green color is a macrophage, and a mesenchymal stromal cell is a red color. We infusion, and we infusion to the carbon tetrachloride induced liver cirrhosis model. And we checking two-photon microscopy. And this is under anesthesia, and we can checking in vivo model. And we can finding what was repopulated, what was happened in this model. And so we can finding, this is very important finding. And this is a macrophage, and this is mesenchymal stromal cell. A black color is a liver and lung and spleen and traceable. And please focus. Bone marrow derived macrophage is day 1, day 3, day 7. Macrophage is repopulated. What is the mesenchymal stromal cell? Day 1, we can finding to liver. What is the day 3, day 7? That is the disappear. And mesenchymal stromal cell, it may be okay to lung, that is repopulated. This is very, very important finding. And please focus. Blue color is a liver fibrosis area, and green color is a macrophage. Okay, oh, sorry. Okay. This is two-photon microscopy, and please focus here. Gradually, liver fibrosis is decreasing this model. And this is very important. And we think the macrophage is very important to improve the liver fibrosis. And please focus here. Mesenchymal stromal cell infusion, that is the majority is depopulated to lung. And a working cell is a macrophage. Macrophage somehow affected it to the, and macrophage is fibrosis regulation and induction of the liver regeneration is occurring. And this is mesenchymal stromal cell and macrophage interaction is very important in this model. And many people knowing is recently macrophage is the pro-inflammation macrophage, anti-inflammation macrophage. So macrophage M1, M2 is important. And our therapy is like immunomodulation. And many people thinking a tissue repair somehow regulated to the macrophage, of course, regulatory T cell and NK cell immune system. And this immunomodulative system is very important. I did it as a several experiment is this study, and deeply checking is my reference, but it's today I focus today's my talk. And my friend, Stuart Hobbes is my friend. He himself is doing is autologous macrophage cell. Yeah, when the starting time is adjusted, the cis paper is autologous bone marrow cell transfusion. One people is thinking transdifferentiation, and one people thinking cell fusion. And Stewart is he himself is macrophage is important at that time. He himself proceeded autologous macrophage therapy or liver cirrhosis. And I myself is a clinical trial using allogeneic mesenchymal stromal cell for decompensated liver cirrhosis model. Firstly, yeah, this is the mesenchymal stromal cell. The important system is the mesenchymal stromal cell low antigenicity. This is very important merit. And we getting is this is I collaborated a lot of company, and I getting adipose derived mesenchymal stromal cell and preparing, and I proceeded with the allogeneic mesenchymal stromal cell therapy. And this is a mesenchymal stromal cell. This is a CD19, CD73, CD105 is a positive cell and cell growth. And tolilinase differentiation, osteogenic, adipogenic, and chondrogenic is occurring. And this is a mesenchymal stromal cell. And other point, important point is MSC and MHC. And this is uh, MSC is MHC class 1 is a positive, but class 2 is a negative. And this is a very important system of the mesenchymal stromal cell. And I also checking is CD8 positive T cell activity to adding is this is ADSC is PBMC interaction 1 times zero point 0.1, 1 times 0.2, 1 times 0.5. And the increasing of the mesenchymal stromal cell and CD8 positive T cell activity is decreasing. This is very important system. And I also, we thinking the macrophage is very important target. So we also checking this experiment. This is mesenchymal stromal cell. This is bone marrow derived macrophage. And we doing a transfer system. And we checking is how macrophage is changing the polarity. So we can finding it. This is by adding to interaction mesenchymal stromal cell and macrophage. It's macrophage shifted. It's a M2 phenotype. This is very important finding that to proceed with the clinical. And other model is this is a carbon tetrachloride injection. We're using is mesenchymal stromal cell, and we checking is a Series threat staining. And this model we can finding is in this model is liver fibrosis is improving this model. And we also using a MASH model also, and this is staining of the liver fibrosis is improving, and we can proceed it in clinically. And this is the 2017. I started the allogeneic adipose tissue-derived mesenchymal stromal cell therapy, CHILD-2B and MASH and HCV origin liver cirrhosis with Roth Pharmaceutical Company. This is just the first Japanese trial. And this is broadcasted with the Roth Pharmaceutical Company and Niigata University collaborated to proceed with allogeneic mesenchymal stromal cell therapy. And this is just our case. And to getting is medical waste to getting is adipose tissue-derived mesenchymal stromal cell. And important point that this phase 1 clinical trial, we didn't using immunosuppressive agent. And this is very important. We infusion from peripheral vein and infusion and checking. And this cell is preparing from company, and this is patient is an infusion. And we did it the phase 1, phase 2. And firstly, we're doing a decompensated. I also doing a clinical 2, phase 2, patient uh, orientated clinical trial or also doing. And we checking decompensated, compensated, and finishing analysis. And now we are preparing a phase 3 study now. And this is my schema. The important point holds that now allogeneic mesenchymal stromal cell. The important point is mesenchymal stromal cell macrophage interaction to improving liver fibrosis to activated the liver regeneration is very important. And we finishing phase 1, phase 2 is a finish. Firstly, we doing a decompensated liver cirrhosis. Now, I also finish a compensated liver cirrhosis. Now, I myself is company is thinking we would be proceeded phase 3 and now is under consideration. Another point, yeah, now I also doing HMGB1 peptide. This is MSC, inducted peptide, and I doing is a Shionogi company. This is a phase 2 compensated liver cirrhosis patient. Now my paper is now revised and will be, the paper will be published in the future. And other part is extracellular vesicle have to proceed it is. That is recently my theme and have to using this new strategy, extracellular vesicle. And many people knowing mesenchymal stromal cell, the size is 10 micrometer. Extracellular vesicle is 100 nanometer. The size is very different. And this is the nanomedicine have to treat the patient. And this is a cell. This is a mesenchymal stromal cell. Secreted, this is extracellular vesicle. And this is the membrane and including DNA, RNA, amino acid, and the size is 100 nanometer. After using, and many people, yeah, I myself, yeah, why? We proceeded to this system, and after using extracellular vesicle is very important. And many people also doing International Society extracellular vesicle. This extracellular vesicle is interacted with this is bacteria derived, blood derived, milk derived, and body derived, and somehow regulated the cell-cell interaction. And have to create it, this nanoparticle is very important. Yeah, to think once again, my finding to photon microscopy and mesenchymal stromal cell is depopulated majority to the lung, but it's liver fibrosis occurring to the in liver, and working cell is a macrophage, and there is some interaction. Mesenchymal stromal cell and secreted extracellular vesicle might be very important. So I proceeded this experiment. And this is our finding is the interferon gamma induced mesenchymal stromal cell derived secreted extracellular vesicle exosome is very important and somehow affected the macrophage. Macrophage shifted M1 to M2. And this is very, very important. Please compare. This is control. This is adding extracellular vesicle. And this is interferon gamma inducted extracellular vesicle. Please compare. Right to panel is the macrophage is moving, is very, very rapidly moving. And activation of the macrophage is occurring to the extracellular vesicle adding system. This is the phagocytic activity. Please focus. This is red color is increasing. And phagocytic activity is also increasing this system. So I also checking. We checking this is the EV and mesenchymal stromal cell inducted EV, and we checking this is anti-inflammation phenotype is activated by extracellular vesicle adding system. And this is very, very important system. And in vivo model have to rethink it. And this is a carbon tetrachloride 8 weeks. And this is a control and mesenchymal stromal cell. And this is interferon gamma inducted mesenchymal stromal cell. And this is EV. And this is interferon gamma inducted EV. Please compare. Here, we can find in vivo model. EV is okay. Mesenchymal stromal cell is okay. EV is okay. And yeah, we ourselves is doing allogenic mesenchymal stromal cell. That is the firstly phase 1, phase 2 clinical trial. Now we're thinking is after doing a phase 3 trial. And the other thing is I also doing is a basic study. EV is improving is liver fibrosis. This is very important finding. And we also checking is this is a CX3, CR1, EGF mice. And this is extrahepatic derived macrophage can be identified by GFP positive cell. This is green color cell. And vehicle, this is EV and interferon gamma inducted mesenchymal stem cell EV. And this is the green cell is increasing. And recruitment macrophage from extrahepatic is occurring in this system. And I also checking a single cell analysis also. This is a single cell analysis. We can finding and this is anti-inflammation macrophages increasing by EV. And this is the regulatory T cell is also activated this system. So, and this is, we published this paper, Nature Publication, Regional Medicine, and this is we can finding is inflammatory gamma mesenchymal stem cell secreted exosome and extracellular vesicle. And this exosome is recruited macrophage, and macrophage is changing anti-inflammation and improving liver fibrosis and activated the liver regeneration. And this is a photo from Vancouver. And my friend is thinking of extracellular vesicle exome form. My friend is Eva Rhodes in she's Austria. She did it the first in human collateral application human stromal cell derived extracellular vesicle therapy, and this is the first in human. And this human study was studied. This is the phase 1 clinical study. And the thinking is have to adapt it. Previously, we doing mesenchymal stromal cell and cell therapy. And important system is have to proceed with the extracellular vesicle therapy, EV and cell source. Yeah, some people is thinking mesenchymal stromal cell, but it's immortalized mesenchymal stromal cell might be important. And indication and which timing, which disease is the most important. And preclinical model is very important. And other point is to checking biodistribution. And we did it is a two-photon microscopy. We can finding is some cell is depopulated to the lung and liver, but is now we also checking is extracellular vesicle and effect and safety. And other part is important to the regulation. This is a new nanomedicine and potency assay. And manufacturing process is very important. Scaling, storage method, sequence, and price. And we can doing to depend on this, we proceeded this clinical trial design is very important. And this is my friend is making is the red color exosome. And this is a tomato system to induct it. This is exosome this is the red color. And we checking biodistribution. And we checking to adding this red color exosome is hepatocyte, hepatic stellate cell, activated stellate cell, and cellular cell. And this cell is not depopulated. capturing is exosome. But macrophage is very easily capturing is exosome. And I think is to thinking is a liver fibrosis, liver cirrhosis patient is macrophage is a key. And thinking of the liver model, and I think is the macrophage is have to changing is anti-inflammation is a key issue. And fortunately, we can finding is red color exosome is the capturing is the macrophage. And we also checking is in vivo model and to photon microscopy system. We can finding is this red color is capturing is the macrophage. And this is very important finding is we publish this paper. And thinking is clinically. This is a two-way. Many people to getting is exosome, ultracentrifuge, and other system is a tangential flow system. And thinking is a large scale. And many people, laboratory based, are making ultracentrifuge system. But other system, tangential flow is a thinking is a clinical. The grade is more adapted. We comparing is ultracentrifuge and a tangential flow system, and we checking this system. And please compare. This is we are 8 weeks is carbon tetrachloride injection and infusion. And this is the tangential flow. This is control, ultracentrifuge. And this both system is available to improving its liver fibrosis. Thinking as a clinically and more large scale is a tangential flow may be okay to proceed it. And recently, many people is focused to the exosome because is now there is no FDA approval exosome drug was not approved. But it's, in the future, this market will be more bigger size and market overviews, market oversize becoming bigger. And many people, of course, many people doing mesenchymal stromal cell, but other people doing is extracellular vesicle is have to adapt it to the human. And this is a segment analysis recently. And target disease, I think it's a fibrotic disease is very good of the extracellular vesicle therapy. And, and this is the condition, clinical trial. Now, phase 3 is especially to the lung disease is proceeded. But there is a not FDA approved exosome drug is not approved yet. And firstly, naive EV, mesenchymal stromal cell-derived EV is proceeded, but in the future, engineering EV will be expected. Many people is trying to do more to studying this field. But it's unfortunately exosome therapy. This is a globally clinically based unregulated administration is becoming is a concern. Yeah, this is many people in Japan is we are concerning because this is we have to taking care because proper way extracellular vesicle therapy will be very big to cure more patient, but is not no good therapy is still there, and so I myself is making Japanese Society Regenerative Medicine, and this is a basic point to consider regarding preparation in the extracellular vesicle and their clinical application in Japan. And this I am also one of the author. And PMDA, and this is a regulation society also making a rule book. And this is the first clinical guidance. This is I chair Japanese Society Regenerative Medicine. And this is the guidance of the clinical application extracellular vesicle. And this is very important. Have to using the clinically this new nano medicine have to create it. I go into the ICEL and 2025 April and ISCT May 10. And this is guidance. Now we are doing, this is mesenchymal stromal cell. and culturing and preparing. And master cell bank is very important. And have to correction, purification, and extracellular vesicle. And other point is important is in vitro and in vivo evaluation and properly animal model and potency assay is very important. And next stage, storage and period condition and regulation is very important. And to proceed to clinical trial is very, very important system. And this system is very, very important to proceed this thing. And I go into the ICEL and ISCT and Japanese Society of Regenerative Medicine making a MOU, International Society Extracellular Vesicle, and 2000 July 20th. And I also a member of the ISCT Exome Committee member. And this is we are now thinking the proper way and international harmonization have to proceed the extracellular vesicle therapy. And other thing is a Japanese Society of Regenerative Medicine is proceeded. This is the database, National Regenerative Medicine database, and this is the DEEP is the Regenerative Medicine evidence accumulated platform. This database is very important to proceed the things. Many people knowing is have to using mesenchymal stromal cell. And the next stage is myself is making a criteria. And Japanese Society Regenerative Medicine making EV guidance. And to making a properly EV product and proceeded with the clinical trial. And clinical database is getting. And we can proceed it morely next stage. And thinking of the liver disease, recently, many people knowing is evaluation of the fibrosis is very much developed. And this is ultrasonography, this is MRI sonography. And to evaluate it, the liver fibrosis is also very important step. And anyway, now is one part is have to treat it EV, and other part is many hepatologists can understanding have to evaluate it is liver fibrosis. And in this condition, I proceeded more. This is the joint meeting. I now proceeded at 2028 Japanese Society of Regenerative Medicine ICT ISL International Conference will be held Kyoto, Yokohama, or Niigata. And now this is proposal thing. And this is now, we are now 2026, and several clinical trials proceeded, and this is a novel therapy. And sometimes with a medicine and diagnosis, oncology, and aging, and this is a global harmonization is very important to proceeding. And today's member is very, very important. And next day we can succeed at this conference. Please join and to come to Japan. And this is my final slide. Eliminate one corner. Together we rise the future of the liver medicine. Thank you for your attention. Thank you very much.
Thank you very much,